This presentation is going to be focusing on the rough endoplasmic reticulum. As shown in Figure 1, the endoplasmic reticulum is located in the eukaryotic cell. The eukaryotic cell is a type of cell that contains a nucleus and organelles that is enclosed by a plasma membrane. This cell belongs to living organisms such as plants, animals, and fungi. Figure 2 shows a close-up of the endoplasmic reticulum. The ER is made up of membranous tubules and flattened sacs known as cisternae. The internal space in the ER is called the cisterol space or the lumen. An important fact to know about the endoplasmic reticulum is that it's an extensive network of membranes that accounts for more than 50% of the total membrane in a eukaryotic cell. The ER plays a critical part in the transportation and production of lipids and proteins. It can be found throughout the cell, but is mainly connected to the nuclear envelope. Because of this, the lumen of the ER connects continuously with the space in the nuclear envelope.
There are two different types of endoplasmic reticulum: the rough endoplasmic reticulum, or RER, and the smooth endoplasmic reticulum, or SER. The main difference between the two is that RER has ribosomes that can be found on the outside of the membrane, while the SER does not. In Figure 1, you can see the ribosomes dotting the rough endoplasmic reticulum. In Figure 2, you can see how the SER looks more tubular than the RER, which looks more flat. Even though there are two types of ER, we are going to focus on the rough endoplasmic reticulum and the role it plays in the cell. The rough endoplasmic reticulum is considered to be the factory role of the cell. Its main job is to either synthesize proteins to be secreted by the cell or to localize the plasma membrane into certain cytoplasmic organelles. This process of processing and packaging proteins is known as protein synthesis. As shown in Figure 1, transcription is the first step in protein synthesis. It starts in the nucleus, more specifically in the DNA. However, the DNA is too important to be moved around, so a copy is made, and the copy is known as messenger ribonucleic acid, or messenger RNA. Then the messenger RNA leaves the nucleus, and the second step takes place. The second step of protein synthesis is translation, and this is shown in Figure 2. After the ribosomes on the rough ER receive the instructions from the messenger RNA, the processing and assembling of proteins starts to take place. This process is known as translation.
During protein synthesis, the proteins that are designated for secretion have a special sequence called the signal sequence that can be found in the first amino acid of the polypeptide chain. The proteins that are synthesized in the ER all have specific endpoints in the organelle. For example, some proteins might stay in the ER, go to the Golgi body, or be sent to lysosomes. Once the synthesis is completed, the synthesis stops until the ribosome is docked at the RER. Once it is docked, the synthesis continues with the new protein threading through the rough ER and is soon cut off from the polypeptide chain. The proteins are moved into the cisternal space where they are folded and are added to carbohydrates to transform them into glycoproteins. After they are turned into glycoproteins, they are shipped off to the Golgi apparatus where they are sorted off into different sections of the cell. If a protein is designated for localization, then the ribosome remains in the cytosol and the completed protein is released into the cytosol.
The rough ER can be found extremely close to the nucleus, which means that the rough ER is able to exert a distinct influence over the processing and packaging of the proteins. Due to their close proximity, the rough ER can quickly send signals over to the nucleus, informing it if possible problems arise in the proteins. If proteins are misfolded and not folded at all, they stand to accumulate in the ER lumen. As a result, unfolded protein response is triggered and causes a reduction in protein synthesis. To summarize, the rough endoplasmic reticulum is a type of endoplasmic reticulum that can be found in a eukaryotic cell. The rough ER gets its name from the ribosomes attached to the membrane, making it look rough under a microscope. Its function in the cell is to act as the factory for proteins, packaging and processing them.
This concludes our presentation. Here are the websites and textbooks we used as sources for our information. Thank you.